Hello, my name is Lexi Nadolski, and in this video I will be covering the history and biology of the West Nile virus. The West Nile virus was first discovered in 1937 in a female patient from the West Nile district of Uganda during a research study that was being conducted on the spread of yellow fever in that region. By the 1950s, it was discovered that West Nile virus was present in Egypt, India, and Israel, and by the early 1960s, it covered Eurasia. The first known case in the Western Hemisphere occurred in 1999 when an outbreak originating in Queens, New York, spread to the neighboring states of New Jersey and Connecticut. The West Nile virus is now endemic to the Eastern Hemisphere and experiences large outbreaks in the warmer months, particularly in Africa. It is beginning to be established in North America and has recently started making appearances in Central and South America. So, what exactly does this viral molecule look like? The West Nile virus is a single strand, positive sense RNA animal virus. The single strand of genetic material is enclosed by the nucleocapsid and is highly prone to mutation. The nucleocapsid is surrounded by an envelope membrane, and on the exterior are envelope proteins that serve as attachment sites to allow the virion to connect to the receptor sites of the potential host cells. Now that we understand the structure, let's take a look at how this virus is transmitted through its replication process. The process of virion replication begins when the envelope proteins on the exterior of the virion attach to the cell receptors of the host cell. The virion is then engulfed by the host cell's membrane and becomes its own floating vesicle entity inside the host cell by the process of endocytosis. The membrane then dissolves, releasing the nucleocapsid and eventually the viral RNA free into the cytoplasm of the host cell. Some of the viral RNA is synthesized into proteins, and the rest of it enters the nucleus and is replicated by undergoing transcription and translation by borrowing the host cell's RNA polymerase. It's at this point where mutation of the virus occurs, as RNA polymerase is highly error-prone and often makes mistakes in replication. The replicated viral RNA and proteins then reconvene at the endoplasmic reticulum and emerge combined in a vesicle at the other end. The viral vesicle then moves toward the Golgi apparatus and assembles into new virions, which head toward the host cell's membrane and are released as new replicated West Nile virions that can spread and infect neighboring cells. Almost all animals that carry the virus are unaffected. Birds are the primary carriers of West Nile virus, and mosquitoes acquire the virus by preying on infected birds. The mosquitoes are the vectors that carry the virus and then transfer it to vertebrate hosts through a bite and into the host's bloodstream. From mosquitoes, West Nile virus can be spread to horses, bats, cats, dogs, squirrels, chipmunks, skunks, alligators, domestic rabbits, and humans. Upon being bitten, the virions initially infect skin cells on the epidermis called keratinocytes near the bite site, and then travel through the bloodstream to lymph nodes so that they can spread through the body's cells and tissues. Mosquitoes make the perfect vector for West Nile virus because their saliva enhances the fruition of the virus in the host vertebrate by accelerating the rate at which virions bind to the host cells and subsequently spread. In humans, only 1 out of every 5 people who have the virus get sick and develop West Nile fever. The other 4 out of 5 people show no symptoms. Symptoms develop 3 to 14 days after infection and range from mild to severe. Those who are symptomatic may experience high fever, headache, perspiration, chills, weakness, fatigue, swollen lymph nodes, joint pain, or gastrointestinal issues. About 1 out of 3 of those symptomatic will develop a bullseye-shaped rash. Less than 1% of cases go into West Nile neuroinvasive disease, which can manifest as encephalitis, meningitis, meningoencephalitis, poliomyelitis, or reversible paralysis. As of today, there is no vaccine or antiviral treatment that has been developed for West Nile virus. Most cases of West Nile fever can be treated with over the counter pain relievers and fever reducers to manage symptoms. Some severe cases may require hospitalization for supportive care, such as rehydrating intravenous fluids and pain medications, especially in cases of West Nile neuroinvasive disease. The best route to avoid the virus in areas where it has run rampant is through preventative measures such as wearing insect repellent, utilizing mosquito nets, and clearing outdoor spaces of standing water. That sums up the history, biology, and current state of the West Nile virus and its resulting condition, West Nile fever. As it's getting warmer out, remember to wear bug spray in highly wooded areas, but in the meantime, West Nile virus is not our biggest concern, so keep washing your hands and social distancing.